We might get started with some of the housekeeping now, and people can just filter in as they like. So good morning, everybody. Thank you for coming to today's seminar. A little bit of standard Zoom housekeeping that I'm sure we're all pretty used to by now. Just please keep yourself on mute until question time comes at the end of the talk, where you can either raise your hand, digitally to signal that you'd like the floor to ask a question. Alternatively, if you want to get in real early, you can type that question into chat. And if for whatever reason you can't speak or your microphone's not working, feel free to type your question into chat and just at the end of it put a little pair of parentheses for me, and I will read the question out for you. Just be quick on the keyboard in case we need some clarification. All right. All right, then I guess we can get started after that. So as we start, I'd like to acknowledge the traditional owners of the land on which we're meeting today, the Wurundjeri people of the Kulin Nation here in Melbourne, the Darawal, Yuin, and Wadi Wadi peoples in the Illawarra, and the Kadagal people of the Eora Nation in Sydney, where the University of Sydney now stands. Today, we're very excited to have Dr. Doyle giving his talk today. He completed his PhD a few years ago at the Research Center for Infectious Diseases at the University of Adelaide, before moving to Bethesda in the US to work at the National Institute for Diabetes and Digestive and Kidney Diseases, where his work received the NIDDK's Director's Award for Individual Research, as well as the Australian Society of Biochemistry and Molecular Biology's Boomerang Award, both in 2020, which is very impressive. Earlier this year, he's moved to the University of Sydney to establish his own lab focusing on the biogenesis of bacterial outer membranes in gram-negative species, with particular interest in the viral assembly machine, which is this really indispensable part of the folding and secretion pathway that's shared by this fantastic diversity of different proteins across different species. So I guess let's get started. If you'd like to share your screen, please.
Oh, thank you very much. I really appreciate that introduction. Where is my share? Is that sharing?
Oh, yep,
Didn't take long to have technical issues, did it?
Oh, it's gone now.
Because I'm not getting a green thing around. All right, you see,
yeah.
see this?
Yeah, we can see the sort of desktop with PowerPoint open.
Excellent.
All good.
Thank you very much for this opportunity to talk, everyone, about some of the work that I've begun over the last few years in the States, and that I'll be continuing at the University of Sydney. And so the key thing that I've just been interested in since I started my PhD is topology of membrane proteins and how membrane proteins are folded. That's kind of my core, I don't know, nerd out thing. And so it's really great to be amongst a bunch of people that have kind of a similar enthusiasm for membrane proteins. So the idea is how do we get, let me put the laser pointer on, there we go. How do we get something that's unfolded, polypeptide that's unfolded into the membrane and folded, okay? And so I like to start with this slide. There's a beautiful hand painting by David Goodsell. And so this is representing a gram-negative cell envelope. And so gram-negative bacteria are diaderms. So we have two membranes, the outer membrane and the inner membrane. And so if we're interested in how the outer membrane is packed with these outer membrane proteins, OMPs, then we have to understand the stages of its synthesis, its secretion across the inner membrane, how it's handled in the periplasm, transferred to the machinery that builds it into the outer membrane, and then how it's released. But also how those proteins, when they're at the outer membrane, interact with all the other factors in the outer membrane, like the lipids, etc., and other proteins. And so what I'd like to just kind of emphasize, and I think you'll kind of on the same page with this, is that we have a big problem now with antimicrobial resistant infections. And so a very recent study has shown that in data from 2019, that at least 5 million deaths per year are occurring that are associated with bacterial infections that are untreatable. And from the leading bacteria are gram negatives, and they can be E. coli and some of these other ones mentioned here. So I think it's pertinent at this point to really understand how this outer membrane is formed, because it is the barrier upon which antibiotics experience difficulty permeating. And two, bacteria have the gram negatives have these amazing pumps. And so as soon as you get past the outer membrane, the antibiotic gets pumped out. So we got this huge problem. So perhaps we'd want to target factors on the cell surface for new antibiotics. And so I'll leave that idea there, and then I'll focus on some of the fundamental things that we're trying to discover. So in case you didn't know this, the gram-negative outer membrane is really weird, okay? So if we take in a live E. coli cell and do AFM, so atomic force microscopy, and zoom in on a patch, you can see these little circles, all right? This is another paper, and they basically have done a similar thing, but they took slothed off outer membrane fractions and did the same thing, allowing them to get a better resolution of what's going on. And all these circles are actually beta barrel outer membrane proteins, integrated beta barrels. So it's packed. It's really packed. In fact, about 50 to 70% of the volume of the outer membrane is protein. The outer membrane is more protein than it is membrane, right? So the outer membrane also has, lipids that you might expect, like PE, PG, and cardiolipin, 70% PE, so this is the main one. But on the 100%, at least in E. coli, 100% of the outer leaflet of the outer membrane is LPS, and this is a bulky molecule, and LPS molecules cross-link with each other across divalent cations. And so you can have OMP interactions between all the OMPs, OMP LPS, and LPS interactions. And basically, all of these interactions cause a very different type of structure, all right? Where you have low diffusion rates. And so basically, it's not a fluid mosaic. You teach kids fluid mosaics, it's not a fluid mosaic. All right, so this is the material that we've got to put huge amounts of proteins into. We got to double that membrane every 15 minutes in E. coli. How do you do that? So that's one of the questions that I'm interested in. So let's just go down a little step and have a look at these barrels further and what do they look like? So in general, they're all anti-parallel beta strands forming a beta sheet folded into a cylinder, and you can see a nice example of OMP here. So, pretty much always in bacteria, you have the first strand pairing with the final strand. The final strand is called the beta signal. And through hydrogen bonding, throughout all of these strands, you get a very stable structure. And in fact, it's resistant to STS. So our native pages are STS pages. And some barrels, you can boil an STS and they won't unfold. So they're rocks, very stable. A tidbit is that they usually have no or few cysteines. And the reason I mention that will become apparent later. Barrels also have these girdles in blue of aromatic residues, kind of to snorkel and orient the barrel correctly in the membrane. And as I said, the C-terminal strand of barrels are highly conserved, and they form these motifs called the beta signal. And usually you have this final phenylalanine at the very end. And it wasn't known what this was for. It's present in at least 70% of barrels in E. coli, but what it was doing was unknown. So if that's the kind of general rules of barrels, then this is the diversity. And you can see just in E. coli, you can have something that's an 8-stranded barrel all the way up to a 26-stranded barrel. So the barrel diameter and complexity can change. So you have differences in the associated in-frame soluble domains as well, periplasmic domains or secreted domains. Here are some of the antibiotic efflux pumps. So you have all of this structural and functional diversity, yet a single complex puts all of those proteins into the outer membrane, and that's the barrel assembly machine. So in E. coli and every species tested thus far, the BAM is essential. So it's you can't knock it out, synthetically lethal, various components of that. So BAM-A in purple here is found in all gram-negative species, so anything that has 2 membranes. And also organelles that are derived from ancient double membrane bacteria, such as mitochondria and chloroplast, also have maintained a BAM throughout evolution. So in E. coli, it's a complex of an integrated BAM-A associated with other accessory proteins. And I'm not going to go into these accessory proteins today, but I'm going to focus on the most important component, subunit, which is BAM-A in purple. So structures had come out as early as 2013, showing what these proteins look like. And immediately we saw that BAM-A is, of course, a barrel itself. So barrels birth barrels. That's nice. I like alliteration. But unusually, BAM-A is not a stable barrel like all other barrels. It's highly unstable. And in fact, there's conformations that were observed where the barrel side, the seam between the first and last strands, open up in the membrane. So what the hell could this be doing? Perhaps this is functionally important, but we really didn't know. And so I guess the main ideas are, how does the BAM take an unfolded OMP, and what are the steps through which it folds it into a barrel, into the outer membrane? And expanding on that, where's the energy come from? So at the outer membrane, we don't have known energy sources. So there's no ATP out there, all right? And there's no useful or known useful chemical gradients through which BAM could harness to help fold proteins. And so I think this question of energy is also something that's very important. All right, so at this point, there are a number of models for how BAM functions to fold OMPs. So an unfolded OMP is synthesized in the cytoplasm, transferred across the inner membrane by the sec translocon. This is an energy-driven process, ATP-driven process. And so it's maintained in an unfolded state. And then once it nears interaction with BAM, perhaps it has some type of proto-barrel structure, not properly folded. And BAM's role is simply to kind of scramble and thin the membrane somehow. And so this could just pop into the membrane because of lowered activation energy. So this is the assisted model. Another leading model at the time that had more BAM involvement was that the reason BAM-A can open up was to be able to present a strand to a newly incoming barrel that's unfolded. Perhaps the beta signal would bind to that strand, and then you'd get threading and insertion of additional strands to build up a barrel on the side that would eventually bud off. But at about this point in time, these models were speculative, very speculative. We didn't know what the folding intermediates looked like, and we still didn't know the energy source. So these are some of the goals: to understand how new OMPs are recognized by folding machines such as BAM, to identify the intermediate stages of OMP folding, what are the energy sources for this, and how does the outer membrane play a role in this process? And this is an often disregarded, frankly, component in the whole process. So to try and understand this, what you need to do is to trap intermediates of protein folding. And this is not an easy thing, because, so firstly, the intermediate stage of a protein folding process needs to be intrinsically unstable, of course, right? Otherwise you'll get falling off into off pathways that are stable, and you won't be able to get out to your folding pathway. So folding intermediates are unstable. So what people have done is to try and make barrel mutants by making strand deletions, loop, all sorts of other insertions and deletions, point mutations. And what they can perhaps do, if you're lucky, is you can get slowed mutants. It's hard to know whether that is an off pathway. Okay, there's no way of testing that. And sometimes, and often actually, they are degraded because they're recognized by other machinery that try and alleviate the problem. So my take on this was, could we do this better? So I started with a type of OMP, which I call fancy OMPs. And these are autotransporter proteins. It's a massive family of OMPs, and it's the most common secretion system in gram-negative bacteria. So it's just a beta barrel protein, just like any other, but it also has an externally secreted soluble domain, which we call the passenger. And the one that I'm depicting here is called ESPP, from enterohemorrhagic E. coli. And we've used this for a long time as a model protein to study what is going on in the assembly process in the outer membrane. So initially it was thought that autotransporters transport themselves, hence the name. So you'd get the barrel inserting into the outer membrane, and then that barrel would act as the pore to secrete from C to N terminus, the passenger, to the external surface. Well, that's all well and good, but it's got a barrel. And what was subsequently shown is that BAM is required for the folding of the barrel. And secondly, we had very good evidence to show that if you somehow slow down the passenger translocation process, then BAM holds onto the old autotransporter until that process is finished. So I took that knowledge to design something new. So if this is the normal autotransporter pathway where BAM-A, at this point, we didn't know what this looks like. This was very hypothetical at the time. BAM-A is holding on to the barrel in gray of the autotransporter. And normally the passenger is traversing the outer membrane. And once it's fully traversed, BAM releases the barrel and you get a mature barrel in the outer membrane. Okay. So what I did was to stop the translocation process by fusing a folded periplasmic protein to the N terminus of the passenger. This is just MBP. So this would fold rapidly in the periplasm and it'd act as a molecular knot to prevent translocation. And then without any mutations to the barrel, you'd have a folding intermediate. That was my hypothesis. We call this MBP ESPP. So I expressed this in bacteria. And what I observed is, so we can track the final stages of protein folding very easy on an STS page. So what I observed was that the barrel folding did not complete. It was completely stable, right? So this was a very highly trapped and stable protein folding intermediate, I thought. So that's fantastic. However, I also designed it such that it would have a small amount of loop exposed to the cell surface. And so what I could do was then cut that, perhaps adding proteinase K to the cells, which would cut that and then release that knot. And then this is all wild type now. So then that could process normally. We can see whether this is an on-pathway intermediate. And in fact, when you add PK to the cells, 100% of the barrels then fully process. So this is the first controllably trapped OMP folding intermediate model without mutating the barrel. So at this point, I wanted to start investigating these intermediates of protein folding. And at that very moment, this fantastic paper came out by researchers in Freiburg, led by Niels Wiedemann. And so I said that BAM is conserved in other organelles such as mitochondria. And so this is a paper in yeast. And so the equivalent system to BAM-A is SAM50. And what they did was they designed an in vitro assay where they purified mitochondria that had expressed versions of SAM50 with single cysteines placed in its first or last strands. And then they synthesized fragments of barrels, also with a single cysteine, and added that to the mitochondria to see what would happen. And so what they were able to see is that they could get cross-links between the first strand of SAM50 and the final strand, the conserved beta signal of the substrate proteins, and also binding to the other side of the barrels. And so what they imagined was that SAM would form a hybrid barrel, a two-sided hybrid barrel with its barrel that it's folding, and then that would bud off. So this was beautiful analysis. And I realized what we could do with my system in bacteria is do exactly the same thing. So what I did was introduce single cysteines on strand 1 of BAM-A in these positions, all barrel lumen facing residues, and likewise barrel lumen facing residues in our stooled ESPP barrel in the final strand beta signal. And lo and behold, if we have the full length here unoxidized, expressed both of those in the same cell, then 100% of the stooled ESPP is bound and cross-linked to BAM-A beta strand 1 through this interaction with the beta signal. And so we could zipper the protein all the way up. So this is a beautiful, specific hybrid sheet. And this is in vivo data. So you can see that very high level cross-linking. So the mitochondrial guys, they could see the substrate barrel binding likewise in a similar fashion to the final strand of SAM50. And so I tried to test that. And you'll have to believe me, I'm not showing the results right here. You can go to the paper. But we didn't see any of that. All right, that was negative data for us. So we didn't see any cross-linking between this strand of BAM-A and the final, the first strand of the substrate protein. Instead, when I placed an outward-facing cysteine right here in the second to last strand, I saw cross-linking along the length of this first strand of the barrel. So now we're on the opposite sides of this hybrid barrel interaction. And weirdly, if you go from the periplasmic side of that strand to the surface of that strand, you get cross-linking along that length with that one position in BAM-A that drops off and then reappears. So, you know, the data are the data, and this is not what we see. We see something like this, and we describe it as an asymmetric hybrid barrel. We got one side where we have the beta signal bound strongly to the first strand of BAM-A, and then this other very unorthodox interaction between the outsides, the lipid-facing sides of the barrels. And also that substrate is swinging against BAM-A. All right. And so we came up with this new model, which I called the swing model. And so I have a little bit of a cartoon here, and I'll explain it as I go. So what we think the reason is for all these different conformations in BAM-A is that the first strand of BAM-A presents to the periplasm, and then perhaps this allows for an unfolded OMP to be bound through its beta signal to that location. And so we've got fantastic data to show that that is the case. And then perhaps you get a sheet that folds, like so, of your building barrel. And then you get a barrel structure. And then the first strand of the client OMP interacts in a kind of very weird way with the C-terminus of BAM-A in at least this conformation and this conformation. And so perhaps this swing allows for integration. All right, that was the controversial conclusion to that paper. And so then it can bud off into the membrane. All right. So, we're kind of getting at some of these, but it's hard to assess, where the energy is coming from, what the membrane's doing, etc. And gosh, it would be good if we could just solve that structure. And so what I resorted to were these smelts. So perhaps you guys know about the styrene malic acid copolymers. So you don't have to use detergents. You just take your native membrane, whack in your polymer. You get these native-nanodiscs, in which lies your protein, affinity purification, and then your protein has never seen detergent. And hopefully you've got the lipids that were bound to the protein in the cell remaining in your discs. And so what I will mention is that I tried all this with detergent and it would not work. And so my intermediates would fall apart. And so I went to this to try and kind of solve that, and it did. So got lucky. I also want to point out that people are really starting to take this up as a great way to study membrane proteins. And so perhaps you should try it out in your systems. So take the bacteria that are expressing BAM and this stored version of ESPP. They express it as a pool of molecules that are trapped in this intermediate stage of hybrid barrel. Take the membranes, add the polymer, get discs, affinity purification, and voila, we got the protein. And so then we started doing Cryo-EM. All right. Here's one of our structures. We solved 10, and here's one of them. It's the highest resolution one. So let's delve into this a little bit more. So in purple is BAM-A, and here's some strands in pink, which is the ESPP that's trapped and being folded. In the other colors are the associated lipoproteins that would be in the protoplasm. And in this region here, you can see the outer membrane nanodisc. Okay. So one area that I'm just going to focus on first is this spot right here that I'm zooming in on. And that is the first strand of BAM-A associated with the final strand, the highly conserved beta signal sequence strand of ESPP. And what you could see is that we didn't need to do expensive Cryo-EM because our in vivo cross-linking was correct, right? So the strands are closely associated with each other in the fashion that we predicted from our cross-linking. But the extra piece of information that was very nice was that the conserved phenylalanine residue at the end of the beta signal was found positioned over a glycine, right? So this basically provided a gap physically for this residue to fold over into. So perhaps this has something to do with recognition of new OMP substrates that need to be folded. I'd also like to point out that this kind of complements very important findings recently of a potential very good antibiotic against gram-negatives that target the BAM. And what they had thought was that this small molecule called darabactin acts kind of like a beta signal. And they solved that structure bound to beta strand 1. And so you can kind of see what it's doing. It's a competitive inhibitor of beta signal binding. All right. So I think these fundamental insights are really telling us how we're going about to target BAM to develop new broad spectrum antibiotics against gram-negatives. But wait on a sec. We can see a few strands of ESPP, but where the hell's the rest of it? ESPP has 12 strands. It should be there somewhere. And so my hypothesis was that in this region of low resolution, this is where the rest of the strands of the barrel were. You can imagine that BAM-A is going around like this, connecting, and then the rest of the hybrid sheet going around. And that the reason we couldn't see that is because the lipoproteins were actually very mobile, right? So we saw a huge conformational heterogeneity in our particles. These are 6 sole structures of the BAM complex from our same sample. And you can see that those lipoproteins and periplasmic subunits are moving around. And what's happening is you're focusing on aligning these areas of the structure, and then you can't see this other independently moving sheet in the membrane. And so my hypothesis was that perhaps we could use focus classification, delete these areas that are uninteresting to us at the moment, and then just focus our classification on this membrane component. And that's what we did, and it was very successful. So here's 1, 2 in yellow, 3 in gray down the bottom, structures of various intermediates that we were able to solve. Again, all from the same sample. All right, so what we were able to see very excitingly was here's BAM-A, and here's the rest of the sheet of ESPP. All right, so you can kind of see this a little bit easier if you rotate the map and slice through it here, and then look down from the cell surface, and you can see BAM-A beta barrel curling around, connecting to ESPP, and here's the rest of the sheet. And in our other intermediates, what we're excitingly able to see was that the folding barrel seemed to fold in on itself. So you can kind of see that here relative to here. And then in what we think is the latest stage, that the barrel barrelizes in on itself towards itself. And so truly this looks like a asymmetric hybrid barrel that I had come up with. So this is kind of a morph to kind of accentuate what we're visualizing here. So we think that the sheet can fold down and in on itself. Okay, so this will play through again. And now we're rotating and looking down on top of the cell. And so we think that it's basically folding in on itself. So those very keen amongst you may have seen something really odd. So in what we think is more earlier stages of barrel folding, where the sheet is more extended, what we're able to see in our outer membrane discs was that the membrane was deflected from the normal plane that you'd expect from the disc. So if we just take BAM-A as the normal plane, and we can see our intermediate sheet, the disc goes up in the air. You know, it's a wild deflection, unbelievable. And so if you compare this against what we think is the later stage of barrel folding, that discrepancy in membrane deflection disappears. And so it's a disc like you would expect, all right? The lipids surrounding it in plane. What the hell is going on? All right, what the hell is going on? So I did a thought experiment. These are the best experiments because you don't have to pay for consumables. So I took some solved structures of E. coli beta barrels. Here's the large one, and here's a small one. And I imagined, let's hold the beta signal strand in plane in the outer membrane in place. And then imagine we're unraveling that sheet in place. And what I saw was in the dark areas, the hydrophobic spanning or the membrane spanning parts of the strands had a discrepancy in its angle relative to its occupancy where the outer membrane should be. All right, there's an antagonism. So perhaps, and this was present in all of the ones that I looked at. So perhaps an idea is that the intermediate stages of barrel folding have evolved to be antagonistic to the plane of the outer membrane. Okay, but that BAM-A's role is to hold on to that strand and even accentuate that angle, that angular discrepancy. And so this is a known angle with the SPP. And perhaps if we extrapolate that to other outer membrane proteins, then you'd see this discrepancy of the hydrophobic area relative to the membrane. All right, well, this is all well and good. Hold that information in your head. Another very important factor is that the outer membrane is different. Remember, I told you about the difference in material structure of the macrostructure of the outer membrane, right? It's very packed. There's lots of interactions. It's non-fluid, it's rigid. And in fact, these guys at Stanford, led by Enrique Haas, who's brilliant, discovered that, in fact, it's not the peptidoglycan, but it's the outer membrane that holds a significant amount of the load of the cell's pressure against osmosis, okay? So, in fact, there is a slight positive elastic potential energy in the outer membrane. It's always under stress, it's always under tension. Okay, put that together. And so what the idea was that if you have intermediate stages of folding that are antagonistic to the outer membrane, but the outer membrane is always under tension, the membrane wants to pull it down. And then the only way to go is to barrelize. Perhaps that's a source of energy for protein folding. And so here's a prop. That wasn't meant to happen. So I do have backups. All right. Take two. All right, you don't need it big because it's on Zoom. So here's the surface of the outer membrane. It's always under tension at homeostasis. But if you have an intermediate of folding that's deflecting the membrane tension is going to want to pull it into plane, okay? Equalizing it, and perhaps that's an energy source for barrel folding. That's the idea. Okay, lots of hand waving. Let's try and test it. Remember, I can track barrel folding in vivo because we have a controllably trapped intermediate. So if you place cells that are expressing this intermediate into culture, LB, grow them normally, and then track barrel folding, the completion of barrel assembly looks like this in black. But if we take those cells and do the same thing, except you transfer them into a hypertonic solution. So cells are going to shrink, spit out the water, and now you're losing your membrane tension. Lo and behold, barrel assembly slows down significantly, but catches up. And so that may be an energy dependent process or defect. So you can do this the other way. You can have a slowed down system, and then during the chase, you swap your bacteria back into normal LB, and then it's going to swell and the outer membrane is going to be tense again. And so that is what happens. So if you just have a relaxed outer membrane, barrel assembly goes like so over time. But if during the chase, you increase the tension in the membrane, barrel assembly increases. And so this was the evidence that we had to support our idea that barrel folding is assisted by the tension in the membrane. And perhaps then there is energy that's useful in the outer membrane. Okay, so this is a summary of these ideas. So we think that BAM-A holds on to these sheets that are intermediates of barrel folding. And so these sheets, these intermediates of barrels have evolved to deflect the membrane when they're not folded properly, all right? And so outer membrane tension is going to pull it back in, and so then that will drive barrel folding faster. And these are just shown here from the cell surface. And so that's a summary of what we found from a fundamental point of view. Of course, there is a lot of remaining questions here. So this is what we observed, our state, but how did it get there in this state? So what were the intermediates prior to this? In the periplasm, how exactly was it first bound? These are open-ended questions that we're going to move on with. Further still, I didn't say anything about the lipoproteins that I'm basically deleting here, but surrounding this intermediate of folding. How do they attribute to the folding process? We really don't know, and that's what we're going to go on with as well. And what I want to finish with in the last couple of slides is to accentuate the importance of working on the BAM complex. And so here's a few papers that have come out recently that focus on finding antibiotics that target the BAM complex. And so Nature Micro, still pretty bloody good. And these are all recent papers that are targeting the BAM complex with small molecules, and they kill pretty much all escape pathogens that are gram negatives. So what I want to say is that the model protein that I'm able to work with as a tool for fundamental investigations is also a very good tool to investigate how these types of new BAM inhibitors function. And so what I've got here is showing you, okay, this is how we know that BAM-A binds to the final strand of ESPP, and here it is here. So we can easily engineer a disulfide bond. And so in normal scenarios where BAM is functioning properly, it'll engage its substrate, and then you'll get a certain level of cross-linking or disulfide bonding, which is basically a readout of binding, all right? But if I take one of those antibiotics, darabactin, from one of those Nature papers, put it on those cells, then there's no binding, all right? And so this is a really good confirmation that this inhibitor is a competitive inhibitor of this site. And this is all in vivo evidence to support these modes of action. And you can take other antibiotics that were postulated, this one in poly4/7 was postulated to bind to the surface of BAM-A. But nevertheless, in our study, in our experiment, we were able to see that binding down here to the substrate was highly inhibited. All right. So perhaps now we have allosteric inhibition. Okay. So this is kind of just to emphasize that we have very good tools to investigate new compounds that target these proteins. And not only this, we can capture and restart folding intermediates in vivo, but in the discs, we can do that as well. So this is the SMALP discs, and so you can cut that in that knot off, and then you can get barrel folding in those discs. And so you can kind of see that data here. So going from here to totally folded barrels. But the crux of this is, well, that perhaps allows us to do time-resolved Cryo-EM to pass out those intermediates more finely, but also to use it as a way of further investigating new antibiotics that target BAM. So with that, I'd like to thank everybody that I was working on with this series of projects, and that I continue to work on now as collaborators. And also I'd like to shamelessly plug that I'm looking for students. And so if you'd like to come to Sydney, if you're a student listening and you'd like to come to Sydney, reach out to me. But also, if you know of great students that want to come to Sydney, send them my way. So thank you very much, and I'll take any questions.
All right, thank you for a fantastic talk. Anybody, if you've got questions, feel free to raise your hand and we'll go through. But while everybody, I think, gets their thoughts in order, oh, hang on, looks like we've got a couple. I just can't see all the hands on the screen. I still might take chair's privilege and ask the first question. So, fantastic talk, thank you. I'm actually a little bit curious about, I suppose, this sort of cell level regulation of BAM-A. So BAM-A as a beta-viral protein, does that facilitate entry of other BAM-A proteins into the outer membrane? Does it have a special chaperone or insertion for, setting up that wider pathway?
Yeah, that's a good question. So BAM-A assembles itself. Okay.
Mm-hmm.
So and we got scooped. So there was a beautiful structure by the Dan Kahn group at Harvard, showing an intermediate of folding of a barrel, and it was BAM folding a new BAM-A. BAM-A assembles itself. So
Okay.
there's an egg scenario. In terms of how many BAMs there are, there's perhaps 2,000 or 3,000 copies of BAM in the outer membrane at any one time. And so that's very, very few if you think about it. If one major porin might have 1 million copies in one cell. BAM-A doesn't have a known unique chaperone for itself, but it does require chaperones for its own assembly, just like other barrels, such as SIR-A, which is a periplasmic chaperone. Yeah, so a bunch of periplasmic chaperones, SIR-A, SCIP, DEG-P. These are all important chaperones in barrel folding. So there is levels of regulation going on, but that hasn't really been passed out yet.
Okay, fantastic.
So at the lowest level, these are regulated by stress response systems, so sigma E. So as soon as you get membrane stress, these systems are turned on as well. So Tom Harvey put out how long ago? It was a couple of years ago, put out a great review describing that the cell is always under stress, and these response pathways are always triggered. And so it's actually, stress at homeostasis is what the cell is under. And so BAM is one of those that's under those controls.
All right, fantastic. We might move on to a question from Isabel, if you want to unmute yourself and ask.
Hi, beautiful work, Matt. Really, really impressive. Well done.
Yeah.
I guess my question is, when you have rearrangement of ESP from the out of plane to the in plane conformation, do you have breaking of hydrogen bond in between the strand of ESP, or do all the hydrogen bonds remain the same?
That's kind of really hard for us to tell, because the modeling there was really difficult. What I would suggest is actually happening, and we kind of see this, is that you have fewer hydrogen bondings earlier relative to a more folded barrel. And it's just because of the structure of the strands relative to each other. As soon as you expand that, you don't really have a stable sheet, right? So that's what's happening. We do see really fun things going on with hydrogen, what we think is happening with hydrogen bonding of the final strand of the substrate of ESPP to the first strand of BAM-A. And so those earlier intermediates have what we think is less hydrogen bonding, more hydrogen bonding relative to the final intermediate. And what we think is going on there is perhaps then you have less energy required for the budding process to release from BAM. So that kind of makes sense as well. So so it's a great question.
So do you think, like, the energy provided by, let's say, your membrane blowing thing, what you showed with the balloon, really correspond to have you tried to align how much energy is coming from membrane.
Have we done math?
Yes.
No. So, it'd be nice to be able to do some type of molecular dynamics in this. I don't know how to approach that. And that's definitely something that's been on my mind, and I'd need to talk to a person that does this. The problem is that you need to design really bespoke systems to accurately or semi-accurately simulate the outer membrane, because you've got LPS there. It all has to be on the outer leaflet. You have to incorporate divalent cations. And so modeling all this, even in a very simple scenario of a folded system, that's a challenge in itself. And people have only started to be able to do this in the last couple of years. Simon Khalid at Oxford is doing these types of things currently. So perhaps I can talk to her, but I think it would be a real long shot to be able to figure this out that way.
Okay, I'm gonna jump in again then. You mentioned that there's relatively few BAM complexes sitting in that outer membrane. Does that mean this is one of these major rate limiting steps for the stress response? Like there's, I guess, a backlog of transcripts ready to go for, antibiotic efflux or like metabolite pickup for the outer membrane. And it's, I suppose, the activity of BAM-A that really limits how much of that's getting into the outer membrane at a time. And if that's so, do you think that could be, I guess, a strategy for developing a new antibiotic therapy? One that disrupts those chaperones while it's stuck in this waiting zone?
Mm-hmm. Yeah, so that's great. So really great idea. So you don't want a backlog of barrels. As soon as you get a backlog of barrels in the periplasm that are waiting to engage the BAM complex, that's toxic. So cells grow slower, or they are almost lethal in some scenarios. Okay. So yes, if you target BAM-A at the highest level, then you're going to get a backlog, and then you're going to get toxicity, and the cell's going to die. But you can also, if you want to go this way, you could perhaps target chaperones, and then you'd have perhaps a similar effect. The problem with that idea is that you always need to get past the outer membrane, right? And as soon as you've got a molecule that is trying to kill the bacteria that's past the outer membrane, pumps can pump it out. All right, there's already pumps there, and it doesn't take much evolution to get a pump to pump out a new molecule, if not immediately without evolution. All right, so what you really want to do is have a molecule that binds to the cell surface somewhere. And so all of those pumps are negligible. They don't do anything. And then you can start targeting all of these escape pathogens. And so this is why people are trying to target BAM-A. I know that it's not just academics that are doing this. Genentech in the States are dumping millions on this, trying to target BAM-A as well. So, but yes, so yeah, definitely, if you cause any type of backlog in substrates, then you've got a big problem in the cell.
Right. Okay.
So yeah, so expression levels of barrels are tightly regulated. Certain members that are slow, you tend to see that they don't have a high abundance, and that kind of makes sense. So yeah, so autotransporters generally are actually quite slowly assembled naturally. Mm-hmm. And general autotransporter proteins, there's only a few thousand copies of those made, relative to very rapidly folding proteins like OMP-A, which you find in 1 million copies or so in the outer membrane.
Right. Okay. And this might be sort of a very basic question, but I thought most of these antibiotic efflux pumps moved stuff from the cytosol back into the extracellular environment, not so much from the periplasm.
I'm pretty sure there's a bunch that pump from the.
I'd be shocked if there wasn't, but I just sort of, I suppose, are these particularly common?
I don't know the answer to that question, but
Fair enough.
my hunch is that yes. I mean, all the reviews, I'm a kind of pictorial person. And so I remember a lot of reviews where you can see these arrows from the periplasm and from the cytoplasm going through these same export bumps.
Right. Okay.
These pole systems.
All right. Thank you. Isabel Doucet, if you want to unmute and ask your question.
Yeah, thanks, Matthew. It was a really, really nice talk and a beautiful work. I may have missed this, but so at what extent the composition of the nanodisk will impact on the formation of those beta barrel? Have you tested composition of the lipids you use in a nanodisk? Have you looked at that?
That's a good question. So let me stop sharing so I can just see me. So with SMAPs, you can't control what lipids are in there.
Okay.
Right? So you just add them to your membrane, and then whatever is retained in the discs is what is retained in the discs.
Okay. Yeah.
So what we did do is lipidomics on our discs, and we saw things that we expected and things that we didn't expect.
Okay. Yeah.
So things that we expected were higher levels of PE lipids. We also saw LPS through a different experiment. But really intriguingly, we saw a huge number of lysolipids. Okay, so single chain lipids with the head still there. So they may act in some way like a detergent or a way to scramble the membrane as well. So that factors in well, I think, with the idea that BAM is very multifunctional. So you're going to disrupt the membrane at the same time as act as a template for folding.
Yeah, because you will need to have a bit of a disruption of the membrane. So that's why I was wondering, yeah, if you, that's great. Yeah.
So, in our preprint,
okay,
which we had to get rid of, we kind of measured coordinates around the circumference of our hybrid barrel to kind of get a gauge of what we could see in our map was the width of the membrane. And lo and behold, where we have protein insertion, we have the membrane thinned. Okay. And that thinning that we observed exactly corresponded to molecular dynamics studies done by others. So big coincidence.
Yeah, great. Thanks a
The
lot.
reviewer didn't like it, so we got rid of it.
No, thanks. That's great.
Okay, it looks like we're through the list of questions for the time being. So thank you so much for coming to talk to us today, Dr. Doyle. Can everybody use your, digital emojis? Give them a hand for a really fantastic talk. The figures were fantastic. And I will see you all later. Thanks, everybody.
Thank you. I really much enjoyed it. Thank you very much, everybody.